So we have the first question about the free radicals. And the thing is, they have unshared electron, and they can bind to anything to complete this pair. And whenever they find this electron, they attack this molecule and take over this electron. So if we take, so let's say you have some kind of molecule, and in the outer shell of this molecule, you have one unpaired electron, which is highly active. So this molecule is very active, and it can bind to anything. So since we have this vacant place, it's not vacant place actually, it's a place that needs to be filled. And this molecule, it's floating inside the stuff, so very close to other molecules. And let's say we have another molecule here, and let's say it's DNA. And DNA, as I said in the previous lecture, is a very stable structure. It doesn't have excess of electrons, etc. But since it's huge, there are many places this molecule can find the electron. So it attacks the DNA. And gets this electron. Okay, so it completes the pair and becomes stable. So free radical gets into a stable condition. And what about DNA? The DNA loses one electron, so minus one electron. And its structures, so if you remember, they are spiral. So we have this spiral structure, and let's say it lost the electron here. And since it lost electron itself, it became very active. So now assume it to be a free radical. It's not free, but it's still a radical, very reactive. So it binds to the nearest molecule, let's say to this place on the other side of the strand. And since it's binding there, these two-stranded DNA, if we unwind it, it should be like this. But now it's more like this. And if we have a gene here, we have a huge problem, because for example, RNA polymerase, it will not be able to get through. So it will get stuck somewhere here. And the transcription and translation of this gene will stop. So this becomes the place for DNA damage. And a lot of other proteins are involved in repairing this region, this gene. And if cell fails to repair it, it will start so-called apoptosis process, which means self-destruction process, and cell dies. So the cell with unrepaired DNA kills itself. Is it clear? They always steal one electron from another place.
Does only one cell die, or starting from that place till the end, DNA dies?
Again, please?
Does only one cell die, or a big part of DNA dies?
So, okay, so just a second. So because this cell, okay, so let's say we have a cell, and the nucleus inside it. So inside this nucleus, it contains DNA. So all these molecules, they go to nuclei, okay? And if this free radical could get into this, let's say free radical is inside this nucleus, and it starts doing its stuff like here, as shown here, okay? And let's say this cell could not repair this DNA. So this DNA inside this nucleus. And this particular cell could not repair it. If it senses unrepaired DNA structures, it sends the signal to kill itself. So this cell dies.
Thank you.
You're welcome. So the question about the tracers and the free radicals can be found in such cases. So there are a lot of antioxidants inside other organisms because they produce it on a daily basis. So we have vegetables, we have, sometimes they say that in wine, for example, or in cheese, they have these natural antioxidants. Also, we produce our antioxidants too, because we usually kind of come across these free radicals. It's not something that happens very rarely. No, it's actually a usual condition for a healthy organism. So even now you're sitting here, your organism works on its basic level, but it produces a lot of these free radicals. And it is needed for some of the reactions, but sometimes these reactions, they fail to finish, and these free radicals, they become kind of free floating inside the cell. So our own organism can handle some of them, but whenever, for example, if we have chemical poisoning or we are being irradiated by some high energies, these free radicals, they are becoming very, very huge numbers. And because of that, we will need to, for example, drink milk, eat vegetables, eat cheese, etc. So this is the product of our cells and the other organisms, because everyone needs the protection against the free radicals. And of course, in tobacco and alcohol, about the alcohol, it actually stops these reactions that have free radicals as players. And because alcohol stops them, these free radicals get free. Okay, so first, let's talk about the secondary conformation. So it was written with a mistake. It's secondary conformation. So again, if you have a primary conformation or primary structure, for example, for DNA, it becomes just one strand with all these A, C, T, G. And you usually work with these primary structures within the course of chemistry. So whenever you see something like this, and you assume that here we have, for example, hydrogens, and here too. So if you write just these 4 carbons, it will be a simple representation of the primary structure. But whenever you show it how it actually forms these clumps and strands in space. It is called the spatial formula. And if you take, for example, these two strands, if you think about, if you want to represent them in a secondary structure, you will need to write them like a spiral, okay? And this will be its secondary structure or secondary conformation. And it is also about the change of shape and active site of the enzymes. That's why they actually sometimes need some activators to release the active center and open the enzyme so it starts doing its job. And radioactive decay. What is this 14? So if you take carbon and it's written 14 here, it means its atomic mass is 14 units. Why it's 14? And how we define the number of neutrons. So atomic mass is equal to the number of protons plus the number of neutrons. We all know that for carbon, the number of protons is 6 because it's the 6th element in the periodic table. And the number of neutrons for the most abundant one is also 6. So if you see something like C12, it means it's the most abundant carbon. If you see C14, it means it has 6 protons and 8 neutrons. So whenever we have the radioactive decay, one of these neutrons, it gives us a proton plus electron plus neutrino. Not really sure if it's the symbol of neutrino. I will look it up. So neutrino and also energy. So this proton stays inside the nucleus. So this nucleus, because of the change of the proton number, this nucleus becomes another element. So from 6 it becomes 7. And you know that the 7th element in the periodic table is nitrogen. And since the number became 7 protons, and this neutron decayed, the number of neutrons will be 7 too. So this is 14 and 14 nitrogen. So about the toxic elements, the thing about them, during the evolution, we evolved, we kind of made a lot of enzymes that make chains. Again, I will need to show it on the screen. Okay, so let's say the first cells on the Earth, they were using enzymes A, B, and C, so that the product of the first enzyme would be would produce the needed substances for enzyme B to produce its product. Okay, so it is the chain of enzymes. So in the first reaction, we have substrate 1 plus substrate 2, and this reaction is controlled by this enzyme, so it produces the product. And for the second enzyme, this product becomes the substrate 1 plus substrate, no, we have S1, S2, so S3 and S4. And this enzyme controls this reaction. So its product becomes S5 for the third one. So plus S6 plus S7, and now we have the final product. So all of these enzymes, they might have some activators or deactivators. So let's say the copper. The copper works as the activator of this enzyme, which means you have some kind of structure, and this place particularly needs copper to change the conformation of the enzyme. Okay, so we have copper here coming to this place, and this enzyme changes its structure. So it, let's say it becomes like this, and we have copper here. And this means that with the change of the conformation, it can accept this, which one? A, okay. So it can take S1 and S2 here, make them closer to each other, and control the reaction of the production of E. So this copper works as an activator, but the problem is this activation center can also take, let's say, plumbum, the lead, and whenever it takes this lead, so again, if this lead comes first, then the conformation didn't change, and this reaction actually didn't happen. So that's why this lead became kind of stopper of this reaction. Of this, oh, sorry, of this whole chain of reactions. And let's say this chain controls the breathing of the organism. So that's why this plumbum kind of becomes the poison, because the organism cannot take oxygen because of only one small molecule. That's why its small concentrations can mess up a whole organism. And now let's talk about the tracers. So the tracers are actually some of these activators. Again, we have some kind of structure that needs this tracer. And let's call this one for this time, which are iron. So this iron needs to come here and activate this molecule. So this molecule changes its conformation only with the attached tracer. So the question about the PET scan. Okay, so the PET scan actually has this, the analog of glucose, this fluorodeoxyglucose. So in this case, we know that this distinct molecule can be kind of signaling to the detector. So we have a detector, and this detector can sense only this molecule. So whichever place this molecule goes to, this detector will see it distinctly. So for example, if it can see the usual glucose as some blank stuff, if it senses this one, it will show a huge focus on the image. The thing about this PET scan, it is very sensitive only to this glucose, for example. And if you take the cancer, it is called the glucose trap because it consumes too much glucose. Most of the times, cancers, they lose ability to take aerobic breathing, so they cannot produce a lot of energy from one glucose. That's why they shift to anaerobic decay of the glucose, the anaerobic energy production. And it is about 38 times worse in the benefits for the cell, which means it needs to take 38 times more glucose. That's why they accumulate all glucose that they find. And this fluorodeoxyglucose is also trapped by the cancers. And because it is easily visible for PET scan, we can see this foci very well on the early stages of the cancer.
How does cancer cells find the glucose? Like, why does glucose accumulate there?
Because they produce a lot of receptors for glucose, and whenever glucose is somewhere near, it kind of catches this glucose and uptakes it. And also it gives the command to the whole organism to send glucose there. So it's usually controlled by, well, let's say we have a new owner of organism, the cancer cell, that actually dictates or enslaves other cells, and these kind of slave cells start working on this cancer. So that's how it happens. So the question about the weakest hydrogen bonds, the problem with these hydrogen bonds, even if they are weaker, they are very, very huge in numbers. So as you could see in the water slide, just think of 1 mole of water. 1 mole of water is 6 times 10 to the power of 23. And this is the huge number. And just imagine that all these water molecules, they produce these hydrogen bonds. And if you take the total energy of these bonds, they of course are bigger than other chemical bonds. Just because of the numbers, you have to multiply the number by the energy of the chemical bond. And this makes it very, very huge. So this pre-biological evolution. So whenever we have these, okay, again. So let's say we have the chemical evolution here, then the biological evolution and biological. So during the chemical evolution, let's say we had only floating elements inside this primary ocean. So everything that that was very simple, CO2, H2O, phosphorus, nitrogen, everything else like sulfur, ferrum, cuprum. So this everything was floating inside this primary ocean. And because of the excess of ultraviolet, some gamma and maybe beta, these molecules were having very high energies, which means they kind of very, very active. So as these, if you remember these free radicals. And what happens next? So the next happens the interaction between these molecules. Let's say these molecules start interacting with each other, And you have to understand that these molecules were very huge in numbers. And since they are very huge in numbers, they form all possible connections, all possible interactions. And this leads to the formation of first, for example, complex organic molecules, CH4 or C6H2O6, etc., etc. So they kind of prepare the whole primary ocean towards the next step of pre-biological evolution. And with the increase of complexity, so whenever we have the complexity increased, it means that we might obtain some new functions of the molecules that were produced with a high complexity. So let's say the sugars started combining with each other, forming, for example, starch or glycogen. And these molecules are very huge, and they have their own conformation, as well as, for example, DNA and proteins. Sorry. So these huge molecules that were prepared from the previous set of reactions, they themselves obtain these catalytic functions and start producing even more complex molecules. They start combining, etc. So whenever we have first proteins and first nucleic acids, we can speak of the formation of these pre-organelles. Let's call them like this. So just a second. So these molecules that proteins and nucleic acids, these first molecules, they could form even higher dimensions of the complexity. And this led to the formation of first, let's call them pre-organelle. And these pre-organelles, they started forming new complexes with each other. That's why we have, for example, ribosome that consists of two distinct parts or units. We have, for example, biomembranes. Biomembranes are very strong in isolating the stuff. So whenever we have a cell, we are usually talking about the biomembrane here. And if we talk about the eukaryotes, we also know that nucleus is also surrounded by the biomembrane. So all these organelles, like ribosomes, the cell wall, the nuclei, they were possible only because of the formation of these very, very complex complexes of the proteins, nucleic acids, and fats, etc. So in the end of this pre-biological evolution period, we have the formation of the first cell. So first cell. And after that, the biological evolution started. Okay, so how radioisotopes occur? I cannot tell you the exact source of the radioisotopes. A lot of stuff happens inside the star. So let's say the core of star starts producing heavy elements. And by heavy, I mean something heavier than helium. And sometimes, since it's inside the star, it can produce a lot of neutrons per proton and make them, let's say, the C14, etc. Or it can be because of the nuclear fission. For example, one uranium, it decays and produces the elements with different numbers of neutrons per protons. And that's why we find these radioisotopes. And what is the reason for changing of neutrons number inside nucleus? Well, you will need to read about the nuclear physics. And it's a very interesting topic. The changes of particles, I mean, how they are related to each other, how they decay, etc., etc. So it's out of the scope of our lecture, but I would recommend that you go and read about neutrons, protons, electrons, at least on Wikipedia. On Wiki, we have a lot of people who can, who devote their own free time to explain very complex ideas with very simple words. So I guess you will need to go there. Okay, so the question about the ozone, yes, it is very unstable molecule, and it happens because, I mean, we have this ozone layer very far from the Earth's surface, and we have it just because the Earth is always bombarded with the ultraviolet light. And because of that, so this ozone molecule looks like this. So 3 atoms of oxygen. So they are in a very unstable condition. So to make this stable, we need to always give it energy from the outside. So we need the outer source of energy for this molecule to just to be produced. And also this, just because this is unstable, the cloud of electrons is shared between all of these oxygens. They all are equally electronegative. So every single atom here kind of tries to pull this cloud of electron on it. That's why we have jumping double bond here. And it jumps from here to here, which means most of the time, we can say that most of the time electrons are kind of located here. They are always everywhere. And that's why we say that these bonds look like this and they are jumping. And whenever we stop this supply with energy, so whenever we stop this supply of energy, this molecule tries to decay, and it usually becomes O2 plus 1 atom, 1 free radical of oxygen. That's why we say that this is the poison, because this oxygen starts attacking everything it sees. So if we don't have this outer source of energy, again, it decays. So happens something like this. So that, just a second, I forgot the question. Okay, so this explains why it's unstable and why it breaks down quickly. So I'm not really sure now the process of production of this ozone. I know that, for example, whenever we have so-called quartz lamps, it produces from the usual oxygen just because of this outer source of energy. So again, we give this molecule the energy to become stable. That's why whenever we have this insulation with these lumps, this oxygen is produced. But if we take the factory production, I cannot say how they do that because I never thought about that. Yes, it is the way that they move. They are actually not moving, so they kind of smashed around the nucleus, and you never know where the electron is at this point of time. You can say the most probable place it might be in, but you can never say where it is now. So if you had chemistry course during your school, you might remember something like 1s2, 2s2, 2p6, 3s2, 3p6. So these orbitals, so for example, s orbitals, as far as I remember, I'm not really sure, so we have nuclei, and they are spherical. All these S2, S, 1s, 2s, 3s, they are spherical and they are closer to the nuclei. So P level, I will need to read about it, but as far as I remember, it's just the thing that was shown in the lecture is just a very simplistic model, and that's why we shown the 8 electrons per orbital. And most of the elements that we work with, they are within this range of orbital completion. That's why we say it on the first level we have maximum 2, on the second maximum 8, and on the third maximum of 6, 8 again. So then, just a second again, what was the question? So each of these levels, they are shells. So energetic levels. And just because, for example, this is the first energy level, and it is, it requires the least energy, it's being filled the first. So this second has the second energy to be on it. Let's say we will never see atoms to be filled with electrons like 1s1, 2s1, because this electron will need much more energy to stay on this orbital. It will jump to this one and form 1s2. And only if there is an outer source of energy again, it can jump to another site, to another orbital, and stay there for some time. And whenever it jumps back, it actually gives some energy. In orbital P, we have the maximum of 6 electrons. On orbital S, we have maximum of 2. If you take the 2s and 2p, the total number of electrons is 8. So again, the first S can only take 2 electrons. The second S can only take 2 electrons, but the second P can take 6. and in total it's 8. And if we take 3s, 3s2 and 3s6, it's also 8.